Song and dance. So welcome everybody to the opening of the summer series of CBR seminars. Pleasure to see so many faces. There are a couple of, I'm Ed Conway, I'm the director of the Center for Blood Research, just in case you don't recognize me. There are just a couple of announcements before I hand over the technology to the engineers amongst us. There is, tell me about what I'm supposed to announce, Parvin, please. CBR Research Day, August the 9th. Registration is up. Be there, okay? It'll be a fantastic day. We're working on getting a fantastic keynote speaker, but most important, all the summer students are presenting and others are presenting along with awards, and there'll be a fantastic afternoon. Second, GAP events. Oh yeah, so the Graduate Awards program for Master's PhD students is an announcement will be coming out shortly in terms of subscribing to that. It's a terrific program that takes place all year. And there's a GAP end of year GAP event coming July the 7th. I'm not sure why I'm actually up here announcing this stuff. Where Parvin should be here. I'm just translating. Anyway, so there's an afternoon. Is that in the afternoon? Or you'll hear more about it. Watch for it. Anything else that I haven't said that I'm supposed to say? Nothing else. Terrific. All right. So here we go. Hong, should I hand it over to you?
Okay. Hi, everyone. So it's great to be here and to start off our summer seminar series. It's a great chance for us to get to know each other and know the kind of work we do and start talking to each other. Okay. So as Ed said, we're an engineering research group. We have not just engineers but other kind of people too. But overall, sort of what we do is we develop technologies for biology and biological research and for life sciences. Okay. And the types of technology we tend to work on are things that are related to cells. And we think of cells as a functional unit that takes information, does some kind of processing on that information, and decides the outcome. And so we want to develop technologies to assay the cell, to look at what the behavior and the function of cells in different ways at the single cell level. Okay. And to do this, we draw on a whole suite of engineering tools. So what's really cool about this is that we have all of this, biology is full of problems, and engineering is full of solutions, and we sort of get to make that marriage in between. So what the types of things that we draw on are like microfabrication, the ability to make small scale structures that are on the size scale of individual cells. Microfluidics, the ability to control fluid flow at very small length scales. Microscopy, there's been tons of advances in microscopy they can draw on. Machine learning, so that's the image, the data processing aspects, and also robotics and automation. Okay, and so for today we have three of our projects that we'll present here. So we'll start off with Emel. So she's going to be presenting on the last part of her PhD, which worked out fantastically. You're always worried about if that last part, the AIM3, whether that's going to work out, but it just turned out great. And so she's going to be talking about, is deformability of red blood cells, is it causal in the circulatory clearance? Because it's not, we know that as red blood cells age, as they circulate, they get more rigid, but does the circulatory system have some way of knowing, how rigid or how soft or how rigid the cell is, and does it have a way of, is it selectively clearing those rigid cells? And so she actually got the answer, which is fantastic. And then Eric will be presenting his work on looking at red cell deformability, red cell quality using blood smears, which is very easy to get. And then finally, Sam will be presenting on the recent work on single cell image cytometry. So I will let Emel take it away. So I'm done here. And come on up, Emel. Just
Okay, awesome. Thanks, everyone, and thank you for the introduction, Hong. So today I'll be talking about the red blood cell deformability as a potential biomarker for the circulatory clearance of the cells in a mouse model. Okay, so red blood cell units, we all know how important they are in transfusion medicine, and we know that when the unit is manufactured, it goes through many rigorous safety checks to make sure that it is as safe as possible. And then the units are manufactured, and then they're considered approximately all the same. Although we do know that these units are not actually all the same. Going to the very beginnings, there are marked donor-to-donor differences that are known. Differences in age, sex, genetic polymorphisms, lifestyle choices. There are a lot of differences that will account for the initial quality of red blood cell units already being not equal to each other. Add to that the effect of storage lesion. So as their cells are stored, they undergo a whole bunch of morphological, biochemical, and metabolic changes, which will reduce the lifespan of the unit. So if you add storage lesion to the already existing donor-to-donor differences, you will end up with different red blood cell units that do not actually perform the same. Then the question is, can we actually measure the differences from unit to unit, and what would we use to measure it? And that's where the red blood cell deformability comes in. So all of the changes that happen within the cell will impact deformability directly or indirectly. And healthy red blood cells are truly the ballerinas of the cell world. They have to be very deformable, and that is vital for their function, for them to be able to deliver oxygen to microcapillaries and the capillary beds. And deformability is affected by natural aging of the cells, by cold storage, donor-to-donor differences. So can we show that reduced deformability of a particular unit actually leads to accelerated clearance of that unit? There are many different techniques in which deformability can be measured. I won't go into too much details. The current clinical standard is ectocytometry. But basically, there are methods where you would look at the deformability as basically one number, as an average of the cells, but you won't know the details about the subpopulations. There are also methods where you will look at the deformability of individual cells, but then you'll have low throughput. What the field of microfluidic techniques brings in, we try to have a look at a large number of cells, but still measure individual deformability of each one of them. And this is where microfluidic ratchet device assay comes in. You can think of it as sort of like a spleen on chip. What we do is we take a red blood cell sample and we push it through a series of constrictions. The constrictions get smaller and smaller in size as you move up the device, from 7.5 microns to only 1.5 microns there. That allows to sort the cells based on how deformable they are, and they'll be collected in individual outlets based on their deformability. From there, you can have a distribution. You can quantify the distribution of cells in each individual outlet, and we have 3 samples here. And if you look at their deformability curve, we have the red curve and the black curves that are fairly similar to each other. And then we have less deformable or more rigid green curve here. So we can analyze this data and compare the samples to each other. Some of our previous work. So first question that we asked ourselves were, okay, so the healthy donor, how different is their deformability on day one? And can we measure this red blood cell deformability between individual healthy donors? And the answer was yes, actually, the deformability signature of different donors is individual. It's different from each other. We also ask ourselves a second question. Okay, so the person A comes, donates now, comes, donates, say, 3 months from now. Will the deformability signature stay the same? And the answer is also yes. So we asked 2 donors, one in blue and one in red, come donate to us at least 6 weeks apart. So most of the cells in circulation would be replaced, to see if the deformability signature actually stays consistent. And the answer to that was yes. The next question was, of course, okay, so if you store cells in the standard blood black units, what do we see? And here as well, we did see, firstly, progressive loss of red blood cell deformability, under standard cold storage conditions. And the magnitude of this change was donor specific. We made a whole bunch of very interesting observations here as well. And I encourage you to have a look at that paper. I won't spend too much time on it here. But basically, the question that remains is, okay, so deformability is individual. There are some donors that are a bit more deformable, some are a bit less. How does it impact the circulation clearance time? So if we look at the two models and two deformability curves, here, the less deformable cells will be cleared out, and only the more deformable ones will stay. Or perhaps all of the cells will get cleared regardless of their deformability status. So which one of them is what we see in an actual organism? And to answer this question, we went to an animal model. So basically what we did is we took mouse samples from the donor mice. We just used regular BL6 for this. We chemically rigidified the cells. So AMT, aminotriazole, is a catalase blocker. What it does is slowly contributes to increased reactive oxidant species formation in a cell without actually killing it. So cells will become a bit more rigid, but they will still keep circulating. Then we will transfuse these AMT-treated more rigid cells and the controls in 1-to-1 ratio. They will be individually labeled so we can tell them apart. We'll inject them into our recipient mice, and we will track the clearance rate of these cells, as well as the deformability profile of these cells over time. So firstly, we show that aminotriazole treatment does make the cells more rigid, and we can see the control here. So aminotriazole treated cells here, same thing over here. We did use very, very low concentration just because we wanted to very slightly rigidify the cells, but not damage them to a point of immediate lysis after transfusion. And then we transfuse them in. So this is a very interesting slide. The cells in green are aminotriazole treated cells before transfusion. So you can see in the red, these are the controls. These are the treated cells. You see that they are more rigid. And then you look at the dashed lines, and that is post-transfusion. All of the cells are gone within first 24 hours post-transfusion. And when you look at the deformability curves of the endogenous cells in black, and then the 24 hours post-transfusion AMT and 24 hours post-transfusion controls, they're almost the same. So the rigid cells actually did get cleared out. This is an average graph, but if we look at individual mice, and we tested 7 of them, you see the same amount of clearance. So all of the rigid cells got cleared within the first 24 hours of circulation. Now, in the first graph here, we're looking at the number of transfused cells over time, and as expected, as the cells circulate, they will eventually get cleared from circulation. This is in 5 weeks of weekly testing. Interestingly, right off the bat, right after transfusion, you see that some of the AMT-treated cells got removed, and those were the rigid cells. If you look at the deformability profile, so these are the rigidity scores of the controls in red, AMT-treated more rigid cells in green, and the endogenous cells, they're pretty much the same between the groups and over time. So the deformability profile of the cells stays pretty consistent between transfused and endogenous. And anything that doesn't fit basically gets selectively cleared. We also looked at the correlation between rigidity scores of the endogenous cells and control, as well as AMT-treated cells, so more rigid cells, and they're very highly correlated. And so far, we see that we can use deformability-based cell sorting to determine the fraction of cleared red blood cells. We see that the less deformable cells are removed from circulation, and we see that the deformability of transfused red blood cells in circulation, at least in our animal model, matches the rigidity score of the recipient. So the transfused cells and the cells that are endogenous have the same deformability profile. Now, the future question that we're trying to address is, so what does make the donor cells circulate for longer? Do they have to be deformable in general, or do they have to match the formability profile, for example, of the recipient that they're in? That's one of the remaining questions that we still have to answer. We are also doing a clinical study now where we are trying to identify the donors that would be able to provide longer circulating red blood cell units to the recipients and to see what role the formability of those units plays. Okay, and a very big thanks to my supervisor committee, and my amazing team, and our amazing emotional support lab dogs. They're very important.
Yeah, maybe there's a question at the end.
Okay.
Okay, cool.
Hello, everyone. My name is Eric, and today I'll be presenting my research on evaluating donor red blood cell quality using blood smears and deep learning. So to motivate this work, I'm using technologies called computer vision and deep learning, and there have been recent high-profile successes for automated object identification and classification. And one notable use of this is in self-driving vehicles. So this is the Cruise self-driving electric vehicle. It is a taxi cab that is operating in San Francisco, Austin, and Phoenix, and it has completed millions of miles of autonomous driving and is actually safer than human drivers for the same amount of miles in the same area. And the way that this technology works is there are cameras and sensors on the vehicle that scan objects around it and identify those objects and determine its position, location, movement, and uses those detections to make decisions for safe driving. And so how this works is you take labeled images for here. This example has cat and dog images. You give it to the model. The model does a bunch of mathematical operations on these pixels of these images, and it detects features that are associated with cats and those that are associated with dogs. And then when it's seen new images of cats and dogs, it can predict which is which. And how that can be important for biology is, let's say you have a whole blood smear shown here, where you have white blood cells, red blood cells, and platelets. You can train the model to identify the features associated with these different types of cells, and then it can predict which cells are present in new images that are provided to it. So the key question that I seek to address is, can this technology assess more subtle image features that are inaccessible to human vision? For example, what about assessing the morphology of red blood cells that is related to their quality? So when I talk about red blood cell quality, I'm talking about it in two senses, in its deformability and also the damage that is induced during storage. And so normal healthy red blood cells have biconcave disc shapes, and then they can be damaged, and this damage is often observed by membrane protrusions. And we see here a progressive morphology degradation, eventually leading to spheroechinocytes. And this morphology progression is observed through natural aging of these cells and also during the course of storage, where the biconcave disc red blood cells are more deformable, they're indicative of less storage damage, and they're of better quality because they're better able to squeeze through the small gaps in the microvasculature. And then the spheroechinocytes are less deformable, they have more storage damage, and are generally of worse quality. So to assess this morphology of the red cells, we use blood smears. This is a clinically standard procedure for assessing red blood cell morphology and is often used for assessing red blood cell pathologies. And so we wanted to use blood smears for the high quality images that they provide, and also for ease of integration into existing clinical workflows. Here's an example of a 60x magnification bright field microscopy image of a blood smear. And so we can see high quality images of the cells and also a variety of different morphologies present in this image. And here's an example of some of those various morphologies observed. We see healthy discoid shapes. We see more echinocyte shapes with membrane protrusions. We see in the bottom right a side view of a red blood cell. And then in the bottom left, we see a small red blood cell that might be a spherocyte. So although we can see all these different morphologies of red blood cells, and these are human observable features, we don't actually know the exact relationship between these morphologies and the quality of the cells. So to get an objective assessment of the quality of these cells, we use a microfluidic device for deformability sorting that Amel discussed in good detail. Here shows a micrograph image at 4x magnification of the microfluidic device. We see red blood cells entering in the bottom right of the bottom right here. They enter into the sorting matrix. They progress in a diagonal fashion through narrow and narrower tapered constrictions until they're output to the deformability outlets here. And so this is a video of the device operating. So the cells are moving through and being squeezed through these smaller and smaller tapered constrictions. And once they reach their minimum constriction limit, where they can no longer pass through those gaps, they proceed horizontally into these deformability outlets. We take images of the cells as they're being sorted to these outlets and characterize their distribution. So here I'll describe my experimental approach. We take red blood cells or whole blood samples from 9 donors and store them for 2 weeks in tubes for accelerated storage damage. We put them through the microfluidic chip to get their deformability profiles. And then we quantify that relationship of the deformability and the progression during storage. In parallel to this, we obtain blood smears, take images of those smears, segment them using computer vision, and then input them through a convolutional neural network. This is the deep learning network that associates the features in the images to the labels associated with those images. And so either we use that for predicting the deformability of the cells or the storage duration. So moving on to the deformability sorting results, we see an example donor here where the week 0 stored cells are the lighter curves and the darker curves are longer times in storage. And we see a general trend of cell rigidification over the course of storage. And this trend was generally seen across our 9 donors. We had 6 out of 9 donors with substantial cell rigidification during the course of storage, with 1 donor depicting minimal storage degradation and 2 donors with cell softening, which this might be characteristics of the donor. It may also be in the the procedure to process these cells. The centrifugation of them might have lysed the fragile cells that would have shown heavy rigidification. And so we use those labels, the deformability labels and the images here to predict the deformability of the red blood cells. So here shows a confusion matrix of 4 different deformability classes. So up here we have the most deformable classes and then the least deformable. So this is the true label of the images and these are the predicted labels. And we see the majority of predictions are along the diagonal, indicating that our model more accurately predicts the correct deformability of the images that are presented to it. Here's an example of images that are sent to the model. And the middle layer, the middle row here shows the saliency maps. Saliency maps depict the pixels in the image that contributed greater to the prediction. And we see that these pixels are all clustered around the cells themselves, indicating the model is in fact learning morphology-based features in the images for predicting deformability. And here we see example images at the 4 different deformability classes. So we have the most deformable on the left and the least deformable on the right. And we see a general trend of more abnormal echinocytic red blood cells in the more rigid cases. So here we show the model predicting 4 distinct classes for deformability. We also wanted to apply the model to output a continuous deformability score. So we have the true deformability on the x-axis and the predicted on the right. And we see a high correlation, Pearson's correlation of 0.71, where more deformable red blood cells are predicted to be more deformable and more rigid ones are predicted to be more rigid. So this gives us good confidence that the model is adaptable and well able to predict deformability from images. We also use this model to predict the storage duration of red blood cells. So we input images that are categorized based on how long they were stored for. again, we generally see more abnormal cells in the later stages of storage. We applied this model for the 9 donors shown here, assessed individually. So we took the data from one donor, trained it, and then took data from that same donor, but that wasn't seen by the model initially, to test it. And we got about 85% accuracy across the 9 donors for predicting how long the blood had been stored. And again, here are the saliency maps, clearly showing that the model is using cell features within the cells for this prediction. And then, so we did show that this model works for individual donors, and then we wanted to use the model, combined all the donors together, and see if it could still predict storage duration. And so we were able to show that that does in fact work. So again, we have the true labels here, the predicted labels on the bottom, and this produced a 78% classification accuracy across the 9 donors for predicting the storage duration. But we also want to see how adaptable this model was to donors that the model had never seen before. So what we did is we took 8 donors, trained the model on it, and on the 9th donor that was held out from training, evaluate if we can determine the storage duration. And so we did that across the 9 donors. And so we got a lower overall average at 61% across these 9 donors, ranging from 75 to 53%. But this is a substantial improvement on random chance guessing, which would be 33%. So we can show here that we have early indications that this model is adaptable enough to associate morphology features in images for predicting storage duration onto new donors that it has never seen before. And this is very important for any sort of integration in a clinical setting, so that the model doesn't have to be fine-tuned and calibrated for each specific donor that it sees. So in summary, I've shown our work using microscope image assessments of blood smears to predict red blood cell quality. We did this using computer vision and deep learning techniques, and we were able to associate red blood cell morphologies with its deformability and the storage damage. And so moving forward, as Amel discussed briefly, we're looking at a clinical study of chronic transfusion patients. And the main thing we are looking at is, can microfluidics, imaging, and machine learning predict the transfusion outcomes based on the biophysical properties of red blood cells? I'd like to thank everyone who made this work possible, and thank you for listening.
One second. Sorry, the computer's being a little slow here. Okay. All right, good afternoon, everybody. Today I'm going to be talking about high throughput single cell screening using nanowell in microwell plates. So we like to call it image cytometry, which is really about doing microscopy. So the advantage of microscopy is that we can retain spatial information about cells over time. And there's a lot of advantages to this over, say, flow cytometry, where you can't really look at behavior such as motion or interactions with other cells. But there's a big problem when you come to talking about high throughput, and that is it's kind of hard to do with a microscope. So over the last 70 years, mainly in drug screening, there's been improvements to microplates to increase the throughput. And that has just been the introduction of more wells. We've gone from 6 to 24 to 96 to 384, and now into these super high density ones, or at least what they consider super high density, 1536 or 3456. But there's a big problem when we get to these density of cells. The amount of fluid in those cells, in those wells, is problematic. evaporation becomes a major consideration and nobody wants to use them. It takes specialized devices and you can't even pipette by hand. And I don't want to use them. And we've kind of, in our lab, come to 3D4 as being the ideal solution where evaporation problems are minimized, and as well as the minimization of plastic in those wells. And I'll get to the why plastic in the wells is problematic in a bit. So let's talk about studying these single cells. How do we do that? Well, if we want to take the method that's true and been done, we take a single cell and we put it in one of these standard wells. But when you look at a microscopy image, where's your cell? How do you even find it? And look at all that wasted space. So there's a new way of trying to deal with this, and that is we introduce nanowells into each of these wells. So we have grids of these tiny little wells that fill each of these larger microwells. And that way we can get a dense number of cells confined to individual containers, but they're going to share the fluid between them. And there are a number of other advantages, such as now every single cell in that single cell study is being introduced to the exact same conditions. So that's how you get the exact same replicate. And that's where we introduce our nano black nanowells. So these offer a glass bottom, both for cell growth and perfect cell imaging. Like there's no alternative that's better than glass. That's why we put glass in our glasses. So using this approach in a 384 well plate, we can get up to 2 million of these nanowells onto a single plate. So when we talk about high density, when we had those 3456, we did 2 million. And this is where I'm going to talk about the materials that go into nanowells, because there are some other options that are available. Everybody here, or maybe the engineers in the room, are familiar with PDMS-based devices. And there's another one, a more commercial product, which is COC, which is sort of a plastic, and it's commonly used in our micro well plates. And the problem is that they only offer moderate quality imaging. It's not better than glass. It's not compatible with adherent cells. So if you look at the red there, basically we call it a single material nanowell. The entire material has to be the same material. So you can't do any treatments to make it compatible with adhesion cells. They'll just kind of crawl out, right? So that's a big problem. And they're also going to have really low contrast for your imaging because you want that transparent material on the bottom, but now the sides are also transparent. And we introduced our solution with Nano Black, where we get super high quality imaging. We're compatible with adherent cells because our walls are non-adherent, and we get high contrast imaging, which is going to simplify our image processing. And these are those nanowells with some cells in them. On the right, we have suspension cells that are well confined, as well as on the left, we have adhesion cells, which grow on the glass surface, they propagate, and they are unable to escape over, as long as we want to keep them. So another consideration here is autofluorescent. Fluorescence is the main way we're going to be making these observations. And so I have two stains here, one in red, one in green. And the thing is, if you look at these, you see the background, you're like, hey, there's some green there, there's some red there. That's because the exposure settings on the microscope were increased super high to try to capture background noise. So the background green you see, that's just through glass. So it's kind of a signal of 0, okay? And relative to that, you can barely even see the nanowell walls. You can see them because they're actually darker than the background color, because we have a fluorescent quencher in the walls, which is going to reduce the signal below background. And that's how we get images that look like this, where we can get beautiful fluorescent imaging across the spectrum, and the nanowells don't interfere. So going back to that compatibility, we do need to make sure that it's not going to interfere with the cell's ability to replicate or any of its other behaviors. And that's where we show here, just using those Raji cells, we can grow them over time. So we can track their proliferation, and we get about 23 hours, which is the proliferation rate of Raji cells. But what I really like is that on the right there, those fluorescent images, those are a proliferation stain which drop in intensity over time as they duplicate. And you can clearly see the drop in fluorescent intensity. However, the signal is still retained because we have really, really good signal to noise ratio. And I talked about contrast a little bit earlier. And What that means is that the walls are much darker than the substrate we're imaging through. And that's important because segmentation is so difficult in single cell imaging. So when you get to data processing, that's where everybody they don't want to look at the data. Like, it's a major headache. And we simplify that procedure just by introducing high contrast in a microwell plate. So you can do the segmentation just doing something as simple as an Otsu threshold. So you can see on the right there, above that box, that's the spectrum of intensities in the image. And if you pick any spot in that blue box, you get good segmentation for extracting the nanowells. And you can see there, very, very quick one-step process. You have all of your nanowell images and you're ready to make your observations. And that's what I like to do. So we do our segmentation and it gives us these brilliant nanowell images like in the top right there, where that's a single cell. We can observe the bright field, we can observe the fluorescent, and we can make a number of observations. And the three different ways that I would do this would be to either look at the intensity or area. Again, that's a very normal way of observing our fluorescent stains. But now we're looking at either a single cell or small number of cells when we look at that mean fluorescent intensity. It's not 10,000 cells. Again, we can do further cell segmentation. This is through glass. We have basically guaranteed we have low cell number. So we've made that segmentation much, much easier. But we can also do what I like to do, which is do outcome prediction based on the nanowells. Now we're going to consider each of our nanowells as a single experiment, which we can do machine learning or some other output prediction based off of. And that comes to my later part of my thesis, which is on assessing CAR T heterogeneity. So CAR T cells are developed to, they're introduced with this receptor onto their surface, which it matches with receptors that are on cancer cells. So if we can fine tune that to the cancer population, the idea is that they'll go in and wipe out the population. And this has been relatively successful in clinical trials and has even become products. But the question we want to answer is, what about the cell population? Is there a superior population of CAR T cells that would be better at killing the cancer cells? And this is important because a massive number of cells is introduced into the patient, and there are a number of side effects which are not so fun. So to assess this, what I do is I isolate the individual CAR T cells so that I have one CAR T cell in each of my nanowells, and I'm going to deposit a number of cancer cells with them. And all I have to do is observe over time and see how many of the cancer cells they're able to kill. And this is sort of my pipeline of doing this analysis once I have those microscopy images where, again, I do that segmentation, which is quite simple. And then I'm going to split my path to identify nanowell locations as well as do the classification on those images. And splitting this into the centroids as well as doing the classification separately just makes it very, very simple for me to be able to observe them over time. I can put them in the incubator, bring them back. It's not a challenge. And what we get out of this is tabulated data that's extremely easy to work with. I can analyze it in Excel and figure out what the population heterogeneity is. So here are some example images from the endpoints of some of my experiments, where we have the cancer cells in blue. We have our CAR T cells, which have been stained red, and then the dead cells, they turn green. And on the right, we can see some of the results, the initial results of heterogeneity from just after 4 hours, in which 50% of the population only kill one or fewer cells. And that's dramatic when we consider that 3 or more, that's 12% of the population is able to kill more than 3 cancer cells. So the big question is, well, what if we develop a way to either improve the CAR T cells or select them prior to patient infusion? Could we develop better outcomes with fewer cells? So I have a couple other projects that are going on in our lab that I'll talk about as well. So the first one here is cell motility, which is being worked on by Pan and Tony. So a common target for cancer drugs is to look at the motility of the cell to target metastasis. And in the left here, we have them stained green, and in the right, we can see the bright field image. And what I really want to highlight here is we can do single cell motility experiments based on bright field or fluorescent. Like the bright field looks brilliant, very, very easy to do that tracking. And they've had very good results so far, looking at TNF alpha as an activator for cell motility before moving on to the next steps. And then we have Jaden here, who is working on insulin secretion. So these are stem cell derived beta cells, and we're looking at the insulin secretion of the individual cells. And he has found that there is a heterogeneous population. And the way this is conducted is by not just depositing cells now, we're going to add ELISA beads, and we're going to throw them all over the wells, and they're going to operate as detectors for the insulin. Okay, so we can see the intensity here, and the important thing here is the intensity of those beads. That's how we measure the secretion. So on the center there, we can see the spatial distribution of the insulin using those beads as detectors. So the spikes, the yellow spike there, that's a cell. And the other yellow spike, that's another cell. And we can see based on that distribution that we get different insulin secretion per cell. And of course, on the far right, you get a much better categorization because this is the distribution just for two cells in 2 nanowells that were close to each other. So we're able to associate insulin secretion in a single well level. And so that brings me to the end. We've developed this micro well plate that we believe is a universal tool that can be adapted to a number of different cell models. And we're ready kind of to give that question. You know, if we gave you this plate, what would you do with it? Thank you for your time.
So we have a bit of time for questions. So we do have questions. So that's 1, 2. Yeah, please use
Audience mics. Oh, yeah. These ones.
So I have 2 questions. The first question is, can you tell us a bit more about how you make these devices, both the nanowells and the separation deformability device? And the other question is about machine learning. So this might have already been done, but hypothetically, say I have some cells where I have a protein stained and it's in the nucleus, and then I treat the cells and the protein is then translocates to the cytoplasm. How easy is it to make an algorithm or machine learning to be able to quantify that?
All right, well, we split it up. There's 3 questions there. So these devices are fabricated in a clean room using a photolithographic process. So basically we use UV light to cure a polymer, which is, you know, we can do at sort of a micron level. So we can make these any size we want. Yeah, can't delve too far in.
For the MICROPLATES Ratchet Device, we also use the same method to have the master of the device, and then we use PTMS, which is type of silicone, to make the single-use versions of it. And for me, each particular assay is one device, and they're single-use. So we always run our assays in duplicates to make sure that the readings are equal. And we've done a lot of calibration with microbeads, with red blood cells and chemically treated cells to make sure that we see the same deformability consistently, and that we see the gradation to more rigid to less rigid cells when we test it.
Yeah. So for the machine learning question, Sam has actually done some work with fluorescent staining of different cell components and has trained a model to, was it 8 different cell types? Yeah, 8 with multiplexed fluorescent dyes, 4 or so dyes, able to differentiate different cell types from each other. So it's definitely possible to, you know, stain protein and then induce drugs to it and then look at those changes. However, the models that we have been using are what are called supervised models, where each image has an associated label to it. So, you know, for us, those labels are deformability or storage duration. For Sam, it was the type of cells. And so the model tunes itself based on the feedback of the label. So it eventually can find an optimal solution for all of its weights and interconnectivity to make those predictions. So if there were external, like if there's another test to evaluate the spreading of that stain into different parts of the cell, very easily a model, even the same model that Sam and I have used, would be able to do that. If there weren't associated external tests to label those cells, you can apply unsupervised learning to it. It just doesn't give as reliable of results, but there are ways around that, and there are ways of adjusting the ways that you do the machine learning to at least get some sort of representation of the different, at least groupings of cells and how they respond. You may just not know what those groupings mean exactly. Hope that answers your question.
Well, I mean, so I guess that like depends on what you're looking for, right? So often what you can do is you can take a picture of the cell and then do an assay on it. And once you've done the assay, you know which cell is which type that you're looking for, like from a fixed perm cell. And then you can go back and associate with that image, then you know what is that, the cell you want or not.
Just to your question about the fabrication, they'll give you a tour. Just down the hall and to the left. All right? Is that true? Yes? In-house? Some of it's in-house.
Yeah.
Most of it.
Most of it's in-house. There you go. All right.
I just have questions about the machine learning.
Can you speak more clearly into the mic? Yeah. That's it.
Oh, there we go. Okay. So when you're doing the deep learning algorithm, is it like, did you start from scratch or is it kind of transfer learning? And I guess the other question I had is, did you see a class imbalance with the different cell morphologies, and does that play into how you optimize your algorithm afterwards? Or was there anything done to address it if it does exist?
Yeah, thank you for those questions. So all the models shown here were trained from scratch. We didn't do any transfer learning. You can pull models off the internet that are pre-trained on a variety of different types of images, say classifying differences between trees and cars, for instance. But and maybe having a pre-trained model as the baseline and then applying ours afterward would improve performance. But since there aren't very many cell-specific databases, there wasn't a lot of examples that would be super useful for transfer learning, but it is something that I have thought about. And then for class imbalances, we do have ways. So we did augment our data sets to make sure that all the classes were balanced, but we didn't have a way of assessing the specific morphologies in each image provided to the model, because we would need to go through those images by hand and classify which morphology they are. And I'm not a hematologist or a blood morphology expert. I would not be able to do that. And it would be very unfair to ask a physician to go through the probably millions of cells that we collected to do that. But we did balance them based on, for the models that we're doing storage duration, the week 0, week 2, week 0, week 1, and week 2, they all had the same number of images provided to the model. And same with the different levels of deformability, the 4 different classes, those were all provided. We would subsample the classes that had many, many images and then oversample the ones that were less representative. So we had balanced classes.
Oh, so I had 2 quick questions. The first of which is for Eric. Leukemias, early detection of leukemias in your samples. Like, is it possible for you to be able to sift through donations from people that might be at risk of developing a leukemia to detect its emergence earlier with the system? It seems pretty well geared towards that.
Yeah, I definitely say that's a possibility. I personally haven't looked at, white blood cells and classifying them. And, I'd be very surprised if a machine learning model couldn't detect a leukemia patient from a non-leukemia patient based on white blood cell images that were provided to it, especially if they were stained with relevant subcellular components. But for early detection, I'd say that's a little bit more of an open question. Could these models determine, cells or donors that are at risk of developing it later on. I'd say that'd be a little bit trickier, but definitely something that could be investigated and has potential.
Yeah, maybe just that a little bit too. So it's really have to think about the numbers because everybody's blood cells look different. Like if we just made an algorithm to see if we can detect differences in people with red blood cells, the outcome would be 100%. Like everybody's red cells look different. So you have to train enough people to then be able to overcome that interperson heterogeneity before you detect any kind of differences that will be.
Okay, thank you. And I guess my other question is, I guess for Sam and Eric, platelets, like the ability to differentiate donor variability in those cells. I imagine they're harder to work with perhaps than the red cells. I mean, you showed PDMS, for example. I imagine the platelets up against PDMS are going to be activated immediately. So I was wondering if you guys had thought about looking at platelets as well as the red cells, and obviously Sam with the white cells.
Yeah, I can address this one. So we've been working with Ellen a little bit in the Divine Lab, and so she has taken platelet smear images, but we haven't gone through the procedure to process them. But there does seem to be potential to evaluate different platelet biochemical factors with the images. The only issue is that platelets are so much smaller than red blood cells or white blood cells. And so we have to image them, we were imaging them at 90x imaging, and it is unclear that even at that high of magnification, we would be able to tell those differences. But it is an area that we have considered, just haven't proceeded all the way through it yet.
Okay, thank you very much.
It's getting there. Good. Okay. Thank you. Eric, it's this question for Eric. You have, it shows some very good data. Prediction is 70 to 80%, 85%. And it kind of like talked about millions of cells you scanned and then got those prediction to be working. So if you are to increase this prediction to 95% or greater than 95%, what is the next things you have to do? Or whether you have to increase the number of cells or whether you have to do something else to increase the predictability.
Yeah, thank you for the question. So, increasing the raw number of cells that we're using, I don't think would give us better results. I've been using close to 10,000 images per class per donor, so kind of on the order of 30 to 40,000 images per donor with roughly 5 to 10 cells per image. So there's lots and lots of cell information that is being provided to the model. But as Dr. Ma mentioned, it's the variability from different donors that is really what we're trying to capture. And so we haven't been able to have a trained model that sees a new donor and give really reliable deformability prediction. It gives an okay deformability prediction. It gives a better storage duration prediction, but it's not reliable enough that we could apply it in clinic right now. So the work that Amel and I are doing on the clinical study, we're going to get tens of donors, maybe even up to, about 30 or so donors at multiple different time points. And that's going to provide us with a wide array of variation between the images. And we think that is the key to getting this model to perform better and more reliably. It may not be the case that we're able to achieve 90+% accuracy, but if we're able to achieve 70 to 80% accuracy on new donors the model has never seen before, that would be a pretty successful outcome.
Yeah. Yeah. So have you looked at 9 donors? Okay. But if you look at Canadian Blood Services, they have a smaller bag comes with the bigger bag. So if you could talk to them, I don't know, so we could maybe get kind of like maybe one slide per each of the bag, because you have already kind of a trained model that could increase the number. If you could get to 10,000 or 1,000 slides, you scan maybe thousands of slides, that might perhaps actually provide an improved prediction.
Yeah, thank you for that.
Can you read it, please? Sorry.
Ask them to. Don, do you want to ask your question, please?
Can you read it? Did you write it out? Oh, you have to turn on.
Is RBC deformity affected by the levels of membrane lipids, phospholipids, and cholesterol?
So if I remember from literature correctly, it does get affected by all of these things, but we don't know to what degree, and we haven't actually measured the deformability versus this particular change versus that particular change that happens in morphology or in the phospholipid bilayer. What we try to do is combine all of the changes that happen and look at one outcome.
Hello? Hello?
Yep.
Yes.
Hi, that's my question. This is Jing Feidong calling from Bloodworks, University of Washington in Seattle. So I'm a little bit far. The question I have is, what happens if I have an increased membrane cholesterol, like patient with hyperlipidemia? Do you expect that the giving force or giving flow rate, the deformity will be reduced or increased?
Yeah. Yeah, I'm thinking. I am not sure. So far, we've been looking at the differences in healthy populations. If you look at the diseased red blood cells, for example, the ones that are infected by malaria, it's actually very easy to detect deformability differences between them of infected versus non-infected cells. The difficulty comes when you look at healthy donors, how different they actually are. So far, we've been focusing on healthy donor population, not as much on the people with particular conditions, although that is a very interesting question, and I would expect to see differences in deformability, but we'll have to measure to see the magnitude and how well we can actually differentiate it with our device.
Last question, Dr. Drysdale.
Thank you, Dr. Conway. The microfluidic ratcheting devices sorting not only by deformability, but by size. And so I'm wondering whether you're taking into account reticulocytes and the couple of % of reticulocytes in these red blood cell bags, because they're larger a little bit, and whether or not diseases that are going to cause fluctuations in the reticulocytes might be predicted here.
Mm-hmm. That's a very good question. We haven't looked at it specifically. What I have done, though, I went into different outlets and I measured from the images, I measured the diameter of cells in different outlets just to see, do I have a lot of very small cells going into outlet 2, which is the smallest pore? And that is not actually the case. I did see some more smaller cells in some samples in outlet 4. And that sort of makes sense because as the cells lose their surface area, they become less deformable. Echinocytes are less deformable, so they will go into more rigid outlets. So I haven't looked at the reticulocytes in particular, but in general, it seems like deformability plays a lot larger role in sorting than just the size itself. Does that sort of answer the question in a way?
It's on. Okay. Well, with that, I would like to thank the Hongma group for a fantastic presentation. All of you for attending from here and from far and wide, from elsewhere in the States and other institutions, hopefully. And we will see you. Well, so let's thank them. We'll see you guys next week, right? Terrific. Thanks again.
Thank you.
Yeah, for sure. Okay.